All right, let's get into this bacteriophage. It is one of the most common types of viruses that we look at and study. Um, it is a virus that attacks a bacteria. That's why it's called a phage, because it's a virus that attacks bacteria. Um, Phages are usually types of viruses that will attack the bacteria. This is a particular type. It's called a T4 bacteriophage. It's the one that kind of looks like underneath the microscope, kind of like an insect. Okay, it has the capsid where the genetic material is contained. It has the tail. And it has an injection site where once it connects or attaches to the bacteria, this is where the genetic material will be inserted into the cell itself. Now, how do these viruses get into a cell? Okay, the plant viruses like the TMV. Okay, it's going to enter through the cell wall. It has to cross through the cell wall to get to the cell. But your animal viruses enter that host cell by a process called endocytosis. Okay, we've talked about endocytosis before. Uh, Pretty straightforward. But for bacterial viruses, like the bacteriophage, it has to pretty much punch a hole into um, the cell wall of the bacteria to be able to inject its genetic material into the cell. So for plant viruses, they have to enter through the cell wall. The animal viruses through endocytosis, and the bacteriophage is going to punch a hole and then inject, kind of like sticking a needle into the cell and injecting its DNA that way. For an animal virus, okay, here's a pictorial description. You have your virus. Here's the capsid and here's the genetic information. Once it enters the cell, uh, usually through endocytosis, okay, the capsid gets broken down. It's just made of proteins, okay, and then the DNA virus can go through two pathways, and we'll talk about those pathways in a minute. But it eventually will get copied uh, into the cell, or the DNA will get copied, and it'll tell the cell to make more copies, and then that's how you get the spread of a virus within an animal cell. Bacteriophage, it has to punch that hole through the cell wall of the bacteria and then inject its DNA. Okay, we talked about that. Here's an actual picture underneath the electron microscope showing bacteriophages literally injecting their DNA into the bacteria. So you can see how there's more than one that's attached to a bacterial cell, and it's going through this process of inserting its DNA. So the two ways that a virus will replicate itself by using the host cell. One is called the lytic cycle. Okay, and in the lytic cycle, you see the bacteriophage attaching itself to the bacteria. It injects its DNA into the bacteria. What happens then is it tells the bacteria to start making copies of itself, making copies of the virus. So it starts to build and copy and replicate the virus, and it produces so many viruses within that bacteria cell that eventually the bacterial cell will burst, and then that releases all of the viruses that were copied inside the cell. Now, what's doing the copying? Remember, the bacteria cell is a living organism. It has processes inside of it that tells that allows the bacteria to make more proteins and to make more copies of itself through asexual reproduction. So what's happening is the viral DNA is taking over those processes and within the bacteria, and then the bacteria is actually making more copies of the virus for itself. So the bacteria is the one that's doing the copying. Okay, that's the lytic cycle. Here's another electron microscope showing you how you can see the line of these bacteriophages that attach to this bacteria you can see how the bacteria had made so many more copies of that virus, it eventually burst, releasing more of those viruses. That's how the lytic cycle works. Okay, here's another description. The virus injects its DNA into the bacteria. That DNA gets incorporated into that DNA from the virus gets incorporated into the DNA of the bacteria. The bacteria then starts making copies of that virus, and it makes copy after copy after copy until eventually that organism cell bursts, and what's released is all of those copies of viruses. And what are those viruses going to do? They're going to move on to another bacteria and attach to that bacteria, and then another one, a different virus, is going to move on to a different bacteria. So that's how viruses spread. That's how they spread and reproduce, or one way. This is called the lytic cycle. So let's look at the lytic cycle a little bit more. Okay, attaches to the DNA, injects its DNA into or attaches to the cell, injects its DNA. I've already talked about that. Then it enters the lytic cycle. New viruses are made by the host cell's resources. Okay, eventually that cell will burst. The process of a cell bursting is called lysing. The cell will lyse or burst, and when that happens, all of those viruses that were made are going to be released where they can go and infect other cells. Okay, that is one cycle of viral reproduction. Another one is this other side of this picture called lysogenic cycle. In the lysogenic cycle, a virus is still going to attach to the host cell. It's still going to inject its DNA. But the DNA is going to become part of the virus. DNA is going to become part of that organism's DNA structure. When that happens, it forms something called a provirus. Okay, so a provirus is formed when the DNA of a virus is integrated into the host DNA and becomes part of that host cell's DNA. The host cell is going to divide normally. So say we're talking about cells that make up the lining of our throat. Okay, for humans. A virus is going to attach to one of those cells. It is going to inject its DNA into one of our throat cells. That DNA from the virus then is going to become incorporated into that DNA of that particular cell, so it's going to become a part of it. It's going to form a provirus. And then our throat cells, they get worn down through eating and breathing and drinking and that type of thing. Well, that cell is just going to divide normally. But as it divides, it's making more throat cells that contain that contain that viral DNA. At some point, there's going to be some type of trigger, some type of chemical trigger or environmental trigger that's going to cause those cells that contain that viral DNA to start copying. And once that happens, they move into the lytic cycle. Okay, so if we go back to this picture, here's a lysogenic cycle. Okay, the DNA of the virus gets inserted into the organism. It eventually becomes incorporated into the virus into the DNA of that organism. So the viral DNA and the cell's DNA become one, structure. That cell continues to divide, nothing's wrong with it yet. It just holds that viral DNA. It's going to make more copies of itself. But eventually there's going to be a trigger. Eventually it's going to be a trigger to where that cell is going to, that virus DNA that's incorporated in that cell is going to say, okay, it's time to start making more copies of the virus. Once that trigger happens, then it moves into the lytic cycle. It makes a bunch of copies. The cell lyses or bursts, and it has produced more of the virus. The lysogenic cycle could almost be like the virus is going into hibernation. Its genetic material is just hanging out, biding its time, and then eventually some trigger causes it to start reproducing. Maybe the virus, we call it, if you remember hearing this when Ebola was on the news not too long ago, We call it an incubation period. Okay? Certain viruses, like the Ebola virus, will infect a cell, and then it'll stay dormant, okay, which means it'll hibernate for a number of days. The Ebola virus has about a 20 to 23 day window where it can stay dormant. It'll stay in this lysogenic cycle where more cells will copy themselves and be produced, which means more virus DNA is going to be made through that process of mitosis. But then after that incubation period, there's something that happens. The cell thinks it has enough to go on, okay, as far as extra cells being made that hold that virus, and then it hits, and then the viral DNA, or then the lytic cycle will start, and that virus is reproduced. All right, now each type of virus can only infect certain types of host cells. Okay, they have those glycoproteins that are going to only be able to identify certain cells. And it's kind of like a lock and key, right? They can only attach to certain cells. That's why the flu virus doesn't show up on our skin. Okay, it's not a virus that affects our skin. It's a virus that actually affects our respiratory cells, like throat, bronchial tubes, those types of things. Okay. The rabies virus, for example, can infect. Well, this leads to this limited host range. This leads to viruses only being able to affect certain types of organisms as well. Okay, so the flu virus. Okay, there's different types of flu viruses, but the flu virus can infect humans, but it might not necessarily affect a mouse. Okay, for example. However, the rabies, this is my example here, the rabies virus can actually infect multiple species. It can infect humans, it can infect dogs, it can infect skunks, it can infect, okay, there's a large array of individuals that virus can infect, but there's a limited host range. And then, like I mentioned earlier, there are certain viruses that are restricted to just certain types of cells within particular species of organisms. The cold virus, if you get a cold, it is a virus. You should not be treating it with antibiotics, but we'll talk about that later. So if you get a cold, it's a virus, and it infects kind of like the flu. It's going to infect your respiratory system. Okay, the AIDS virus, it's going to attack your white blood cells. That's going to affect your immune system. That's all the AIDS virus attacks. The HPV, the human papillomavirus, infects cells of the cervix. So the HPV virus is really just going to affect females because females are who have a cervix. The herpes 2 virus is going to infect nerve cells found within the genitalia. So certain viruses, oftentimes, there's going to be if it's a virus, it's going to infect just a limited type of cell within a limited group of organisms.